Well, hello everybody, and welcome to those of you in the room and also to those of you watching online. It's really good to see that essentially every seat in the theatre is taken. So it's a pleasure to introduce this inaugural lecture to be given by my colleague and friend, Professor, it sounds strange, David Greensmith. Let's actually begin by introducing myself. I'm David Eisner from down the road at the University of Manchester, where I'm Professor of Cardiac Physiology. It's great to see all David's friends, those going back to his school and college days, but I hope you won't mind if I concentrate upon welcoming his family, who are really our special guests today. First of all, Dave's family, Robert, Diane, Ian and Laura, then his wife Christine and her family, Jeff, Catherine, Zena and Stephen, and last but most certainly not least, our special guest of honour, the youngest member, I think, of the audience, Andrew Greensmith. Andrew, do you want to take a bow for us? Wave to the audience. Thank you very much. So I first met Dave, I think, in 2005. He just graduated from the University of Salford and came to the University of Manchester to do a PhD with Mahesh Nirmalan and me. It's particularly good to see Mahesh here today. From the moment he started, Dave impressed us both with his tremendous enthusiasm for the science. He didn't just do what his supervisors suggested, but found novel techniques and approaches by himself. He's also, and it may come in useful for some of you, one of the best people I've ever met when it comes to fixing bits of equipment. He did, however, have some peculiarities. I remember Dave, this is starting to sound like a best man speech. Yeah. I remember him coming to the lab one day with a plastic carrier bag wrapped around one of his trainers on his feet. I asked him why it was there. In response, he looked at me as if I was a complete idiot in a rather pitying way and explained it was obvious. It was raining and there was a hole in his trainer. Perhaps the best part of academic life, to be more serious for a moment, is watching one's students develop and mature. And it's a privilege to have watched Dave grow up over the last 20 years, from the student whom we knew originally to today's professor and associate dean. After finishing his PhD, Dave did a postdoc with us, but it was clearly time for him to fly the nest, and he took up the offer of a lectureship here in Salford. Here he has been successful in establishing his own research group and has attracted more than £1 million in funding to do this. He's also, importantly, put great efforts into teaching. This has been recognised in Salford by the Vice-Chancellor's Distinguished Teaching Award and nationally in 2023 by the very prestigious award from the Physiological Society of the Otto Hutter Teaching Prize and Lecture. He's also clearly done great things for Salford in the international arena, where he's developed new partnerships and globalised student recruitment. He's also contributed to academic life in the UK and internationally as president of the British Society for Cardiovascular Research and associate editor for current research in physiology. Last but not least, he finds the time to be scientific director of the charity KidsCan. All in all, a very busy man. I want to end by saying something about Dave as a person. He's one of the most fundamentally decent people I've ever met, who will always go out of his way to help others. It's a real privilege to have had him as a student and now to call him a valued friend. With no more ado, I'd like to welcome Professor David Greensmith to deliver his inaugural lecture entitled Calcium Measurement in the Heart, the How and the Why. We look forward to it, Dave.
Thank you, David. Hey, so thank you. So welcome, everybody. First and foremost, thanks ever so much, David. That was an incredibly kind introduction. I'm very touched indeed. And thanks to everyone in the room. It's a real honour and privilege to see so many friends here today to come and watch this lecture. And I'm really very grateful to you all. So today's lecture's titled Calcium Measurement in the Heart. I'm going to tell you how and why we do this. I'm going to break it into three parts. The first is really just a bit of a journey, telling you about how I got to where I am now. And then I'm going to move into more of a public lecture, and I'm going to hopefully provide an accessible overview of the heart and the importance of calcium. And that will hopefully provide a foundation to then go on to just tell you a little bit about some of the contributions we've made to the field at Salford with my collaborators. So we'll start at the beginning, my journey. So here's the obligatory baby picture. Thanks to my mum for digging this out. Born 2nd of March 1983. Took a very early interest in biology, as you can see here. But my mum tells me that my first career aspiration was to be a shepherd. This was after a starring role in the Nativity there. I attended St Mary's, then St Luke's Primary School in Loughton near Wigan. And like any good northerner, Brodie, I learnt a brass instrument and played in a brass band. And I also engaged in many other wholesome activities as a child. I have a sister, Laura Greensmith, and we always got along like a house on fire. But joking aside, I've got very fond memories of my childhood, and it really was a wonderful time. So as all children, I was asked this constantly: what do you want to be when you grow up, David? And my answer was always this: a scientist. And really, this is where my first thank you comes in, because in this regard, I've got an awful lot to thank my parents, my dad, Robert Greensmith, my mum, Diane Cartwright, who were incredibly supportive parents and had a very, very strong educational ethos that was always instilled in us from the outset. Now, of course, my mum was a teacher, so education came naturally, but I've got her to thank for all sorts of contraband learning materials that would appear miraculously in my bedroom. And my dad was a biologist by background, and on Saturdays we would do all sorts of fun experiments in the garage, which I'm afraid Helen weren't risk assessed. But nonetheless, I've got an awful lot to thank my mum and dad for. So in 1994, I arrived at Goulburn High School. Not an awful lot to say here, other than my interest in science continued to develop, as did a pretty dodgy haircut. In 1999, I attended Winstanley College, and I took A-levels in chemistry, biology and geography. And actually here, my academic performance dropped somewhat. Now, my parents would cry idleness, which I think, to be fair, there was probably an element of that, being a typical sort of 18-year-old lad. But actually, in my defence, I would counter-argue the element of aimlessness there as well. Now, as I mentioned, from the moment I could talk until this point, I always wanted to be a scientist. The problem is I didn't know what that meant. And as I'll go on to a bit later, science and science-based careers are still very much misrepresented or underrepresented in high school and colleges. And my problem is, and it's still the case to this day, I can't work hard unless there's a point to it. I just can't, there's something about it, I just can't work in a futile way. So probably a bit of idleness for sure, but certainly this played a part. But I can't deny that I usually have my social life on my mind, and that certainly didn't help at all. But I did meet these guys who are in the audience today. This is Owen Griffiths and Jenny Eccles, and they're still the very best of friends to this day. So things changed considerably in 2001 when I arrived at the University of Salford, and I did a BSc in Biological Sciences. And these early days were absolutely transformative, because for the first time, in all that time I've been interested in science, I saw what real scientists were. I saw how you became a scientist. I understood what research meant, the sort of things you see on adverts and tellies for charities, etc. It was going on in the labs, and it was just like a veil had been lifted. And from that point, I never looked back, and I really threw myself into my studies. So this was my bedroom at the time in Eddie Coleman Court. The younger students in the audience will be absolutely appalled at our terrible living conditions. We had to rely on these things called books. There was no such thing as ChatGTP. We had these awful clunky computers, no Facebook, no Twitter. We had dodgy chat rooms. Age, sex, location, anyone? We had no WhatsApp. We had to rely on these old phones and this terrible technology called SMS. We had these massive televisions with only 5 channels. The MP3s were just coming in, but it was all a little bit dodgy. So our music was recorded onto CDs, which we had to put into a machine. But my God, was that music good. And I miss those days. So when I started at Salford, my early aspirations were actually to be a plant biologist. And here are some early photographs of some field trips, which I remember incredibly fondly. And it's also where I met another one of my best friends, Peter McGee, who's also, I'm very pleased, to join us today. But things started to shift in my second year when I undertook some physiology modules at Salford, Systems Physiology 1 and 2, and I thought, oh, this is all right. And I really started to gain an interest in physiology. And that was really crystallised in my placement year. I went off to the University Hospital Aintree in Liverpool, where I did a placement for the full year. And my time was split between two roles. 2 days a week, I worked as a medical technical officer, working with patients doing respiratory tests. But the other 3 days, I got involved in all sorts of really interesting research projects. And it did two things. It absolutely crystallised my love for physiology, but also research. So when I joined final year, I chose as many physiology modules as I possibly could and graduated with a biological sciences degree, but with a heavy physiology bias. So was it all work and no play? Well, believe it or not, this was Salford University's motto at the time. They actually used this on television adverts: work hard, play harder. So of course, being an excellent student, I availed myself. I did what I was told. And this took the form of extracurricular activities, so the debriefs, as my lecturers used to call them after field trips. Chomping around the clubs of Manchester with these reprobates, Fifth Ave and 42s mainly, great times. And of course, the Crescent played a leading role in my education, and I shudder to think how many pints of hides I drunk over the years in the Crescent. And of course, romance blossomed, because it was here where I met my now wife, Christine. To cut a long story short, it was right at the end of our degree, and I bumped into her on the night out, and she came over and recognised me as that tall bloke on the corridor, and the rest was history. So did all this work? Well, I like to think so. In 2005, I graduated from first class in biological sciences, and I was also really privileged to win the Institute of Biology Award for performance. So that was a very proud day. Certainly for me, but especially for my nana. My nana passed away recently, but this was an incredibly proud day, and she talked about this for many years afterwards. So I just want to take this opportunity now to thank those academics and lecturers who really meant to me at the time. You really were inspirational. You put me on a track to essentially research and where I am now. Absolutely phenomenal bunch of people. Again, you really did pull the veil from over my eyes. And I just want to acknowledge Dave Collins, who also passed away a few years ago, and I still miss Dave very much indeed. And actually, as Associate Dean for International Development, now and again, it's me on the stage reading out those names. And I always have a little smile to myself when I'm sitting there and cast my mind back to 20 years ago when it was myself walking across the stage. And that's a very proud moment indeed. So during all this, did I develop an aim? Was there a point to life? Well, academic life. Well, absolutely. It was to secure a PhD. Okay, that became very clear very early on in my degree, and I had no idea where to start at all. It was just something that wasn't discussed, certainly in college. I'm still very grateful to Professor David Story, who at the time was head of our school, and he very kindly accepted a meeting request and went through the nuts and bolts. And he suggested having a chat with Jeremy Allen, who was a new lecturer at the time, and Salford had what was called the Graduate Teaching Assistantship Scheme, which provided PhDs. And this was great because it meant I got to do my PhD and I got to stay at Salford. But obviously, the chances of getting these are fairly low. So I actually applied to the Manchester Strategic Studentship Scheme. And I'm afraid to say, Mahesh and David, at that time, Manchester was my backup option. It was there as an insurance choice. However, Mahesh, my application came across Mahesh's desk, and during this sort of process where all this was going on, and he invited me in for a chat, and I went over, and I was just awestruck. The science that they were doing in the labs at Manchester was absolutely right up my street, exactly what I wanted to do, and the equipment they were using absolutely called to my inner geek. And as David said, it allowed me to play with all sorts of really expensive and nice toys. So I was really fortunate to be offered both of these PhDs, but it was with a very heavy heart that I had to leave Salford behind temporarily, as it turned out, and head to Manchester to take up that opportunity. So there I am. I arrived in Manchester in 2005 and undertook my PhD with Professor Mahesh Nirmalan and Professor David Eisner. And my project was really focusing on heart failure in sepsis. And what we were doing is adding certain immune system messengers, things called cytokines and reactive oxygen species that control the autoimmune system. And we were trying to find out how these messengers altered heart cell function. I also developed I got into programming and developed some new analytical processes, and we still use those to this day, which again makes me quite proud. Now, during my PhD, I went to a conference in America, Biophysics 2008, and my colleague Mark Richards and I discovered a bar called the Yard House, which, as the evidence suggests, served these yards of beer. And we were so thrilled by this. We took as much opportunity to be in the Yard House as possible. We had a great time. Now, Mahesh was so concerned about this. On our return to Manchester, he actually had a word with David and Andy and said, I think Mark and Dave have got a bit of a problem. Mahesh said, don't worry, he'll grow out of it. Not sure how, but... But I want to just point something out. So in my first week at Manchester, on the first Friday, Mahesh called me to my first important lab meeting, which was in the Hog's Head. This was what I witnessed at our first social event, and this was our first conference. So I rest my case. So nonetheless, I graduated in 2009 with a PhD from the University of Manchester, another incredibly proud day for my family and myself, and Kristen and I were going nice and strongly at this point, so that was a lovely day for the two of us, and we look back at it fondly. So what came next? Well, I stayed at Manchester for another 6 years and did a couple of postdoc positions under the supervision of Professors David Eisner, Andrew Trafford, and Catherine Dibb. And I was really fortunate to join a really fantastic lab group of incredible scientists, many of whom became very, very good friends. Now, during this time, I contributed to a number of projects that all really sought to better understand the cellular basis of heart disease. Well, this was my favourite paper. Most credit really needs to go to Gina Galli, who developed a new technique to measure calcium in cells. And I was able to employ that technique to basically settle a debate that had been raging in the scientific literature for several years as to whether or not something was capable of controlling heart cell function. And I think it's fair to say that we put that one to bed with these experiments. And so proud of me was I of these experiments that when I left Manchester, David Eisner put my favourite thing on a mug and a t-shirt. So that was quite good. I'll still use that to this day. So another slide to say some thank yous. So the time at Manchester was absolutely inspirational, transformative. My supervisors, Mahesh Nirmalan, David Eisner, Catherine Dibb, Andrew Trafford over the years, world-leading scientists, absolutely full of encouragement and support. Can't thank them enough. And what was also fantastic is they gave me that sort of academic freedom. When I came up with wild ideas and ideas for new experiments, they always supported that and basically let me get on with it. A couple of other thank yous. Luigi Bertucci really took me under his wing from the moment and was always a fantastic source of advice and support. And the same goes for Stephen O'Neill, who sadly passed away several years ago. And also a special thanks to Vaskin Ohanian, who again sadly passed away a few years ago. But he was kind of, and he wouldn't like me for saying this, but a pastoral mentor. We would go for weekly meetings in his office and talk about anything other than the heart, usually Weber barbecues or high-end hi-fi or photography. So he was a great friend. But nonetheless, this time came to leave Manchester. So I did this because I returned back to Salford in 2014 as a lecturer in biomedical science. And now for the first time, I had to split my attentions between research and teaching. But if you'll permit me, I'd just like to go off on a slight tangent and talk about teaching for a little bit for two reasons. First of all, I'm very fond of teaching. It's a very rewarding part of my sort of academic life. But also, if there's any newer members of academic staff in the audience, I hope to show that teaching and research don't need to be a tension. You can do both and you can enjoy both and you can hopefully have impact in both. So I did a couple of things as a lecturer at Salford. I was a module leader for a couple of physiology modules, proud to have been shortlisted for an award for our COVID adapted delivery of certain practicals. And as programme leader for human biology and infectious diseases, at the time, Salford was actually ranked 9th in the UK for a physiology-based degree. And I also had a little bit of external influence. I contributed to a government report that was scrutinising the way GCSE mathematics was delivered. And for the Physiological Society, I did a couple of webinars on systematic reviews, and I believe that these are still hold record downloads. But I'm most proud of my contributions to widening participation and research. So, dry mouth, one sec. So, why do we need widening participation and outreach at university? Well, around 60% of our students are from Greater Manchester, and the significance of this is Greater Manchester has one of the highest concentrations of deprived localities in the UK. Why that's important? Well, if you look at any index of social deprivation, we learn that learners from socially disadvantaged backgrounds are less likely to progress to higher education than those from relatively advantaged backgrounds. And you can use any index of that to make that point. So why is that the case? Well, I think it's simple. It's because you don't know what you don't know. We know from statistics that learners from disadvantaged backgrounds are often first in their family to attend university. They're less likely to have connections to, in this case, science or equivalent careers. They're less likely to be from families with knowledge of those associated careers, and they're less likely to have access to informal learning, museum visits, etc., outreach events, and robust career support. The take-home message here is that learners from disadvantaged backgrounds are less likely to have insight required to make informed career choices. So what did we do about this? Well, we set up the Salford Schools Network, and this was really a platform to run several outreach events in the local community. And with a great team of staff, we went out to these schools and we ran all sorts of activities. But really the reason was to raise awareness of, in our case, STEM-based careers and higher education-based pathways. Now, what I want to stress is this wasn't to present those pathways as superior. Anyone who knows me will tell you that I don't believe in that. Higher education and associated careers are just one of many, many options open to the young learner. What we wanted to do is simply make sure their choices were informed and that they didn't not enter a higher education and STEM-based pathway simply because they were completely unaware that those pathways existed. And thanks to Christine here, who was absolutely instrumental in setting up that Salford network and also the outreach schemes. And I'm really proud that Christine and I actually managed to get a paper out a few months ago, which reports some of the impact that those events had. So, problem solved, right? We've got more kids going to university. Well, unfortunately, not quite. Now, if we consider the ideal career progression pipeline, at some point, a learner will develop an aspiration to enter a certain career. The context here is STEM, but that could be anything. So they will embark on an appropriate learning pathway. In the ideal world, from the very start of this journey, they'll start to develop graduate capital. And graduate capital is just all the other stuff other than your degree or qualifications that make you employable. Insight, experience, professionalism, etc. And of course, that allows you to graduate with a good degree or whatever you've done and have a great CV, so you're competitive. But we know that disadvantaged learners are A, more likely to consider higher education later on in life, and B, tend to be more unaware of career options. What this means is by the time, even if they get into university, by the time they graduate, they're sort of behind the graduate capital curve, so to speak. So they're less competitive, despite the fact that in almost every case that I've come across, their academic abilities are absolutely equal. So how did we address this? Well, really all credit to Sarah Namvar, who's back there. Sarah set up a whole series of initiatives at Salford that all sought to do one thing, to help students build graduate capital. And we published a few really nice papers if you're interested in this. So just as an example of many of the initiatives that Sarah led, we set up some career mentorship schemes. So these were working groups, if you like, that help students who developed an interest in a certain career get into those careers. So for example, Sarah Nirashni ran one that helped students get into medicine and physician's associate. Tamino looked after our biomedical scientists, and so on and so forth. So how did I contribute to this? Well, I set up the Research Careers Working Group, which was aimed at students who developed an interest in research. And this is because, as my story, for many students, university is the first time that they meet real researchers and understand the pathways required to enter research-based careers. And it's only then that they develop those aspirations to enter research-based careers. So this is not just a problem associated with relatively disadvantaged learners. Students from relatively advantaged backgrounds face the same problem because science and research-based careers are usually completely ignored until people get to university. But the problem is, you see, is the next steps required, usually embarking on a PhD or equivalent, are fiercely competitive. So what the Research Careers Working Group sought to do was first of all provide rapid insight, get them aware of those career pathways from the moment they joined Salford, and then during their time here, help them to build experience through things like placements, projects, conference attendance, you name it, and then to mentor them through that PhD process, application process, like the application and the interviews, etc. Now, I've got to say Matthew Jones has now taken this over from myself because I'm quite my time's quite torn, and he's doing a far better job of it than I ever did. But I'm very proud of some of the impact that we had in those early days, and we had a lot of fantastic undergraduate students who made it into some really stunning postgraduate positions, and I like to think that the Research Careers Working Group helped out. So I was really proud of the fact that I won a couple of awards in recognition of this contribution to widening participation and outreach. The 2019 Vice Chancellor's Distinguished Teaching Award and the Physiological Society's Otto Hutter Teaching Prize and Lecture in 2003. And another thank you slide. Back then, I belonged to a teaching leadership team that I think was an incredibly effective outfit. Under the leadership of Professor Mahesh Nirmalan, who was the subject head for biomedicine at the time, I worked alongside Sarah and Lucy Smythe, who were also programme leaders, and we did an awful lot. It was a bit, how do I put it? It wasn't part of our day job, put it that way. You know, we developed an awful lot of schemes, and I think we worked very well as a team. And I do miss those days. They were great times. So anyway, back to 2014. While all this was going on, of course, as a lecturer, you're expected to develop an independent research profile. So where did I start? Well, the first thing I did was hire a van, and I think on one of these raids, my mum helped me out, and I pinched some equipment from Manchester, and in combination with a couple of small startup grants, I managed to eke out the beginnings of the lab in the corner of a room in the Cockcroft building. I was then very fortunate to recruit Natasha Haggraft, who's here with us today, and she was an exceptional my first PhD student, and an exceptional scientist, and also a very kind individual. And she's now doing fantastically well for herself at Manchester Metropolitan University. But the point is, we started to produce some data. And then we were asked by Luigi Venatucci at Manchester and Philip Eaton, who was then at King's College in London, to contribute to a project that we're working on. And what this meant is that one of my first outputs out of Salford actually went into Nature Communications. So where did this lead? Well, it certainly gave me the confidence and a little bit more academic clout, so to speak, to apply for some other grants and facilitate funding. And from that point, really, the cardiovascular physiology lab at Salford has grown quite considerably. And now we've got all sorts of really cool equipment that we use on a daily basis. And in terms of equipment, we really are comparable to any of the bigger labs that cardiovascular research labs that you might find around the world. We're probably just a little bit more scaled, but we've certainly got the ability to generate that high quality data. But of course, equipment is absolutely useless without people. So did my lab group grow? Well, absolutely, and I'm really proud of the fact that over the years, there's always been a lab group with a given critical mass. Now, people come and people go, but there's always been a really fantastic team of scientists working on our projects, and I really do owe all these guys an awful lot because that their commitment and passion for the research has been inspiring to see. So then in 2022, things changed a little bit for me at Salford. So during my lectureship, Nirmalan asked me to help out on some international development projects. We were trying to set up a partnership in Sri Lanka. And this really gave me the bug for international development. I then took on a directorate leadership role, which was lead for international, and that bug started to grow. Now in 2022, this position came up, Associate Dean for International Development, and I thought, well, I'll throw my hat into the ring. Never thought for 1 minute I get it, but I did. So here I am now doing international development with a fantastic team of people who I'm really flattered to have come here today. So my day job now is really focusing on the development of international partnerships for the school, instilling a sense of global citizenship into our students, and also growing international recruitment. But despite the advice, I do still teach because I absolutely love it, and I certainly don't want to give up all my teaching. But I guess in the context of today's lecture, am I still research active? Well, I'd like to think so. The lab group's as strong as ever. This is the current lineup. I'm now very happy that Matthew Jones is now senior lecturer at Salford, and he's certainly develops himself as an independent investigator. So Matt and I now co-lead the Cardiovascular Research group here at Salford. I was appointed as president of the British Society for Cardiovascular Research in 2025, and in the same year appointed as scientific director of KidsCan. So yes, although absolutely love my day job, so to speak, I'm still absolutely research active. So while all this was going on, a couple of life events. In 2016, I got married to Christine, finally, after quite some time. And who's this, Andrew? Yes, little Andrew came along in 2020, who's the apple of our eye. So that's the end of part one. So I'm just going to change tack a little bit now and give you more of a public lecture, hopefully an accessible overview of the role of calcium in the heart. And this will hopefully form a platform to then explain some of the contributions that we make at Salford. So as you're probably aware, the heart is a pump. And it's pumped because it generates the force required to pump blood around your circulations. Now that force is generated by heart muscle, which we also call the myocardium. So what forms heart muscle? Well, specialist cells called heart muscle cells, we properly refer to those as cardiac myocytes. And your heart contains around 2 to 3 billion of these cardiac myocytes. Now these cells are incredibly specialized and complicated cells. So today I just want to focus on a few key features. So the first things I'd like you to remember is that, as with all cells, membrane, i.e. the sarcolemma in this context, that the cells are membrane bound. We call the membrane the sarcolemma. And the membrane separates the inside of the cell, which we call the cytoplasm, from the outside space. We also have an intracellular calcium store. This is a large store of calcium found inside the cell. We call this the sarcoplasmic reticulum, or SR. We also have these structures called sarcomeres. We don't need to get too bogged down into these today, but they're basically formed by what we call contractile proteins. And sarcomeres can change their geometry, and that shortens the cell. Now, each of these cells is a discrete force generating unit. So how do heart cells generate force? Well, it's because they're contractile. So here's a video of a real heart cell, and we can see that it's capable of contracting, i.e. getting shorter, and then relaxing, i.e. getting longer on every single heartbeat. So what coordinates this cell contraction? Well, it's a process known as cardiac excitation contraction coupling, which is simply the sequence of events that takes the excitation of a heart cell right the way through to its contraction. So to understand this, we need to understand how heart cells are excited in the first place, and then what those coupling events are. So let's start at the beginning with excitation. So if we just consider for a moment the beating heart and what that requires to be useful. Well, first of all, those contractions must be involuntary. It would be useless if we had to think or will our heart to contract. Secondly, that contraction must be rhythmic, so it can act as a pump, and it must be perpetual, because obviously if the heart was ever to stop contracting, we'd die. So what permits this? Perhaps it's the nervous system. It stands to reason. Nerves are responsible for exciting most other structures in our body. Well, in the heart, it's absolutely not the case. Because hearts are what we call myogenic, in that they have an intrinsic ability to generate contractile function. And this is demonstrated by this experiment where a real heart's been removed from a body, cannulated, and this cannula is simply providing all the things that heart needs to contract. It's basically fake blood, if you like. But what I want you to consider here is that any nerves that used to innervate this heart have now been completely severed. And yet there the heart is perfectly happily beating away. And it will continue to do so for many hours if looked after. So where does this myogenicity originate? It's in a structure known as the sinoatrial node, which you may be familiar with as the cardiac pacemaker. And what the cardiac pacemaker does is basically, on every heartbeat, it generates a wave of excitation. And you can think of this as an electrical signal. And that electrical signal propagates through the heart muscle, and when it hits a given cell in that heart muscle, it excites that cell. So what happens when a cell is excited? Well, it fires what we call an action potential. And an action potential is simply a temporary increase in membrane voltage. So here's a real action potential from a real cell. We see that at rest, the cell is held at about -80 units of voltage, and that soars to around 40 units of voltage, millivolts. But that action potential, that lasts for about half a second. So that action potential triggers contraction. Yeah, so that action potential is basically what triggers contraction. So that's excitation taken care of. What we need to ask next is what are the sequence of events that couple that excitation to contraction? Well, it's a calcium cycle, in that in every cell and on every single heartbeat, excitation causes calcium inside the cell to rise. That causes the cell to contract, and because the cells contract, the heart contracts and the chambers eject blood into the circulation. Calcium levels are then allowed to fall. That means the cells relax, the chambers relax, and so they fill with blood. So what this calcium is, it's a perpetual cycle that drives that constant contracting and relaxing of the heart cell on every heartbeat. So the next question is, what produces this rise and fall of calcium? Well, it's a set of specialized structures in the cell called the calcium handling proteins. So remember, here's our sarcoplasmic reticulum. That's a calcium store inside the cell. The first protein I want to draw your attention to is the voltage-gated calcium channel. These sit on the membrane and they open to allow calcium to enter the cell. Also on the membrane are calcium exchanges, and these pump calcium outside of the cell. On the surface of the sarcoplasmic reticulum are SR calcium release channels, which we also call ryanodine receptors. And these open to allow calcium to enter the cell from the calcium store. And we also have these SR calcium pumps, known as the sarcoendoplasmic reticulum calcium ATPases. Thankfully, we shortened that to SERCA. And these pump calcium back from the cytoplasm back into the SR. And remember these guys, the sarcomeres, complicated little fellas, but I just want you to remember one thing, is that when calcium binds to sarcomeres, they shorten, and when the sarcomeres shorten, the cell shortens. So how do these proteins cooperate to produce the calcium cycle? Well, to understand this, we can start the journey, if you like, at the point where the cell's relaxed, pre-excitation. And the cells is relaxed because cytoplasmic calcium levels, intracellular calcium levels inside the cell are low. And what this means is you have very low binding of calcium to these sarcomeres, and so the sarcomeres, therefore cell, remain at a resting length. At the same time, calcium inside the SR is found at a very high concentration. However, that calcium can't gain access to the cytoplasm because we have a membrane in the way and calcium can't flow over membranes. So the question becomes, how does SR calcium gain access to these sarcomeres to produce contraction? Well, during contraction, we have excitation. And as we saw earlier, during this excitatory period, an action potential fires and the membrane's voltage increases. Now that increase in voltage is sensed by these voltage-gated calcium channels on the surface of the cell, and they open to let a small amount of calcium into the cytoplasm. Now, that amount of calcium is too small to cause sarcomere binding and therefore contraction. Rather, in a process known as calcium-induced calcium release, this trigger calcium, as we call it, binds to the SR calcium release channels, which causes them to open and release a much greater amount of calcium into the cytoplasm. Calcium levels are then increased to levels that are sufficient to allow calcium to bind to these sarcomeres, so the sarcomeres shorten and therefore the cell contracts. So how does the cell relax? Well, it stands to reason that to allow relaxation, we need to restore those resting calcium levels. We need to remove the calcium that's just entered during excitation or following excitation. And in heart cells, there are two fundamental processes that achieve this. The first are those SR membrane pumps, circa, and the second are these calcium exchanges on the cell surface. And these two proteins work in concert to remove the calcium that entered during excitation, restore resting calcium levels. Calcium then detaches itself from the sarcomeres re-lengthen, thus the cell relaxes. So how do we describe this rise and fall of calcium? Well, experimentally, we record it as the calcium transient. And the calcium transient is simply the change of cell calcium over the duration of one heartbeat inside the heart cell. And there are two components of this that I want to draw your attention to. The first is the peak calcium level, and this is what causes the cell to contract. The other is the resting calcium level, and it's this level that permits relaxation. So what each calcium cycle does is provide a perpetual series of calcium transients, as you can see here. And those calcium transients over your entire life drive cell, therefore heart contraction. So is the calcium transient adaptable, or is it an all or nothing event? Well, absolutely, because what your heart cells can do is modulate those peak calcium levels. So here we've got calcium transients of different amplitudes and higher peak calcium levels. What does this do? It allows the cell to modulate contractile force. So here's a really nice relationship. I love these beautiful physiological relationships, and it shows the relationship between peak calcium level and force of cell contraction. And we see that as we increase peak calcium levels, the heart cell beats harder. It contracts more forcefully. So how does the cell modulate peak calcium levels? It does this by modulating SR calcium content. And here's another beautiful physiological relationship that was described by David. And this shows the relationship between the amount of calcium in the SR and peak calcium levels. And we see that as we increase SR calcium content, the amount of calcium released during the calcium cycle increases. But this isn't a simple relationship. It's what we call a cubic relationship. What that means in simple terms is you only need a very, very small change of SR calcium content to produce a very, very large change in peak calcium level. So modulation of SR calcium content is the main physiological way that the cell modulates the peak level of calcium during the calcium cycle, then cell force, therefore heart contraction. Why is this useful? Well, it gives your body a way to control cardiac output. The cardiac output is simply a measure of your heart's overall activity. It's simply the amount of blood your heart pumps per minute. And at rest, that volume of blood is around 5 litres. But during periods of, for example, exercise, we might need to increase cardiac output manyfold to say 20 litres a minute. And by increasing SR calcium content, the cell will beat harder, and therefore the heart will contract more forcefully, and it will eject more blood into the circulation per heartbeat, and it also beats faster. And the converse is true during periods of rest. So the take-home message here is that heart function is dependent on cell contractility. Contraction of the cell is driven by the calcium transient, And anything that changes SR calcium content will alter heart cell function. So we've just seen an example of that. In health, we use this relationship to physiologically modify heart cell function, but sometimes drugs or disease will also modify SR calcium content. And if you change SR calcium content, you'll change the peak calcium transient, and therefore you'll alter cell contraction. So to better understand the cellular basis of both health and disease, scientists need ways to measure cell activity. So how do we measure cell calcium levels? Well, we use molecules known as calcium-sensitive fluorophores. And fluorophores are simply a type of molecule that emit light when they're excited by light. And that's the property we refer to as fluorescence. Now, the cool thing about calcium-sensitive fluorophores is their fluorescence, i.e. their brightness, is proportional to the amount of calcium in their environment. In other words, the higher the concentration of calcium, the brighter they will glow. So we can essentially use light to measure calcium in cells. So how do we do this? Well, we take individual heart cells and we load them with these fluorophores. Fluorophores accumulate in the cytoplasm, and then we use a high intensity light source to excite the cells at a given wavelength, in this case, a blue light. And when we do that, those fluorophores will emit light at different wavelength, in this case, green light. And we can collect that green light and turn it into a digital signal using all sorts of fancy equipment. And if we look at a real experiment here, this is a cell that's been loaded with one of these fluorophores, and you can see that preceding each contraction, if you look at the end, it gets shorter, we have a flash of light, and that flash of light is due to the rise of calcium. So we can sort of backwards calculate the actual calcium concentrations from these light flashes. So can we record even more information? Well, yes, because we can simultaneously use a technique called patch clamping. And patch clamping is where you simply, it's quite tricky. You add a small microelectrode onto the surface of the cell. Now, this cartoon here kind of betrays the complexity of this, because what we need to remember is heart cells are absolutely tiny. They're only about 0.1 millimeters in length. So these microelectrodes are minuscule and require quite a lot of elaborate equipment to get in position. But nonetheless, if we're successful, patch clamping allows us to measure things like those membrane voltages, the entry and exit of calcium across the membrane, and also the amount of calcium that's actually contained in the sarcoplasmic reticulum. So finally, what about contraction? We want to be able to see what these heart cells are actually doing from a mechanical perspective. And to do that, we use special cameras which record at a very high speed, and a technique called video detection. And basically all this is doing is picking up the shape of the cell. So when the cell contracts, that shape is tracked by the system and generates these contractility transients, which basically shows the extent to which heart cells shorten, then re-lengthen during a twitch. So how do we employ these techniques? So that just leads me to the final part of my talk today, which is just a bit of an overview of our collaborative contributions to the field at Salford. These are all the sorts of experiments we do here in the labs at Salford. So all our work basically revolves around this central point, which is to better understand the cellular basis of heart function in health and dysfunction in disease. So just some previous projects. Worked with Luigi Ventucci and David over at Manchester to better understand the regulation of cell calcium in health. Also with Luigi, Philip Eaton at King's College and Andrew Trafford to see how the body regulates cardiac output. Natasha graduated from her PhD a few years ago, and again, huge thanks, because Natasha was my first PhD student, and she carried on my work that I started at Salford looking at heart failure in sepsis and produced a lot of nice data. I also dabbled in a few experiments with Eisner and Andrew Trafford, looking at Eisner is a parasitologist and she's interested in some new antimalarials, and we had a look at how those affect heart cells. So moving on to our current projects, what are we actually doing at the moment? Well, one avenue of discovery is looking at new drugs. So this really started with Matthew Jones. I managed to get some industry funding to fund an industry PhD, And Matt looked at the potential of certain natural products, plants basically, as new anti-cancer and cardiac drugs. And Matt's now taken this line of investigation as an independent stream. I still work with Javier Palicos in Chile, and again, we're interested in other natural products, and we're very keen to understand whether these can be used as new ways to treat high blood pressure. Beth Ann Samfire-Noden is another fantastic PhD student in my lab at the moment. She's in collaboration with Professor Nidal Bittar at Blackpool Victoria Hospital and Sarah Withers. We're trying to better understand the cellular basis of coronary artery disease. And the other main focus is cardio-oncology, which I'll come on to in a moment. But again, a huge thanks to Amy Foster, who was another incredibly talented PhD student, graduated a couple of years ago, and she produced some really nice data. And we're at the moment, we're trying to get that to the next phase. But in the meantime, we've got Alicia Staley and Charles Middleton, two other fantastic PhD students who have taken on where Amy left off and are really doing some great work in understanding this cardio-oncological process. Now, I don't have time to go into all these, which you'll be glad to hear. So I'm just going to focus on one example, which is cardio-oncology. So what is cardio-oncology? Well, it's basically the understanding of how anti-cancer drugs affect the heart. Now, we've got a problem when it comes to cancer treatment. Cancer drugs have never been better. Cancer survival rates are now better than they've ever been because we've got some fantastic drugs. The problem is many of those drugs cause heart failure. So today's cancer survivors becomes tomorrow's heart failure patients. And it's not really clear why that's the case. So our focus is a type of anti-cancer drug called the anthracycline. Now, anthracyclines work by generating really potent molecules called reactive oxygen species. And these are just very damaging molecules that kill cells. And they do this by inducing lethal oxidative stress in those cancer cells. Think of them like bullets. Now, we know from the literature that anthracyclines cause heart failure. People who have been treated with anthracyclines, who survive cancer, have a much higher chance of developing heart failure later in life. What isn't clear is to the extent to which there's a cellular basis for this, and also whether oxidative stress contributes to that. So how did we approach answering some of these questions? Well, Amy used an example anthracycline called doxorubicin, and she exposed this to heart cells to measure the effect of doxorubicin on calcium handling, contractility, and oxidative stress. So the first question we asked is, does doxorubicin alter the calcium transient? Well, here's an experiment where we've got some nice calcium transients under control conditions, in other words, before we have the drug, and we see we have nice regular calcium transients of a nice amplitude. What happens when we add doxorubicin? We get a profound decrease in the amplitude of calcium. And this is also reflected by the mean data. And from this, we can take that, yes, this anti-cancer drug does decrease peak calcium levels. Now, remember I told you earlier that peak calcium dictates the force at which the cell contracts. So does doxorubicin also reduce cell contractility? Well, absolutely. We get a mirrored profound decrease in contractility. That's also supported by the mean data. So we can take from this that, yes, doxorubicin does decrease peak calcium levels, which causes a decrease in cell shortening. So why does peak calcium decrease? Well, remember this relationship that we looked at earlier, in that peak calcium is heavily dependent on SR calcium content, because you only need a small change in that content to produce a large change in peak calcium. Because of this relationship, in the lab, if ever we see a decrease in the peak calcium level, the SR calcium content is the first place we look. So does doxorubicin decrease SR calcium content? Well, we can measure this experimentally by adding caffeine to cells. What caffeine does is it locks open those calcium release channels on the rhydine receptor. And that basically causes the SR to empty itself of calcium. If you've loaded the cell with a calcium-sensitive fluorophore, that manifests itself as a caffeine-evoked calcium transient. And the take-home message is the amplitude of that caffeine-evoked calcium transient gives you an indication of how much calcium there was in the SR. When we add doxorubicin, the magnitude is significantly decreased. So on average, what we can take from this is, yes, doxorubicin does decrease SR calcium content. So our next question was, are these effects dependent on oxidative stress? Is oxidative stress causing this decrease in SR calcium content? The first question here is, does doxorubicin actually increase oxidative stress in these cells? So Amy did this beautiful experiment where she loaded cells with a different type of fluorophore. This was an oxidative stress sensitive fluorophore, and in the same way that calcium sensitive fluorophores increase in fluorescence when calcium increases, these increase in fluorescence when oxidative stress increases. So we're just using light to measure oxidative stress. And what we see is doxorubicin produces a concentration dependent increase in those oxidative stress levels. So of course, this doesn't necessarily mean that that's responsible for the changes to calcium cycling. So to probe this, what Amy did is repeated the experiments we saw earlier in the presence of a drug called N-acetylcysteine. And N-acetylcysteine is just a very powerful antioxidant. It basically removes oxidative stress from the equation. And if we compare the effects of doxorubicin in the presence of N-acetylcysteine with those in the absence of N-acetylcysteine, we see that there's a significant attenuation in the effect of doxorubicin. And what this tells us is that, with a bit of squinting, around 50% of the effects of doxorubicin appear to be dependent on oxidative stress. So, yes, we know that anthracyclines cause heart failure, but hopefully today I've convinced you that there's certainly a contributing cellular basis to this, and it seems that oxidative stress does play a role. Now, there's much more to this story than meets the eye, and Amy and I are trying our absolute best to get this into a publication, which hopefully will be out soon. And of course, as I mentioned earlier, sorry, the story absolutely continues with these guys. So that's it. My final slide is some thank yous. So first of all, to the funders over the years, without their generous support, these experiments simply wouldn't be possible. So huge thank you. Also to my collaborators around the world. I owe all these guys a great deal. And to my family, of course, who are always an absolute source of inspiration and support. So from left to right, most importantly, we've got Audrey, which is Laura's dog. My sister Laura, my stepdad Ian, my mum Diane, Kathy, my mother-in-law, the boy Andrew, Jeff, my father-in-law, my dad, father, Robert, Christine, and here's me in the middle of cooking a turkey. And also to a fantastic brother-in-law and sister-in-law, Steve and Zena. And between Laura, Steve and Zena, they also make some fantastic world-leading aunties and uncles. And finally, and by no means least, Christine and Andrew, who really are the absolute rock in all this, constant source of support, make it all worthwhile, and put up with all the traveling, all the late days at work, all the etc. You can imagine comes with sort of juggling all these different commitments. So I really can't thank you enough. You really are fantastic. And that's it. Thank you. And any questions?
I think you've left a couple of minutes for questions. I don't know if there are questions from people in the room or from those online. Got a question, Andrew?
All right. Dinosaur hearts. Not a great deal. I presume they had hearts, and they were probably very big. I think Robin might be the person to advise on that. But yeah, absolutely. Hearts as a organ, they evolved very, very early on in the divergence of species. So dinosaurs certainly had hearts, and they would have been just as important to dinosaurs as they are to us. Great question, Andrew.
We've got a question here from, oh, sorry.
Yeah.
When you wash that out,
yeah,
the long-term effect.
It's a great question.
Just for people online, probably can't hear the question. For those online, the question is, when you get the doxorubicin effect, when you wash off the doxorubicin, do you see a long-term effect?
So the short answer is yes. In the interest of simplicity, I missed the wash part of our experiment off, but we do a washout, and what we see is a partial return of function, but not across the board. So some of the effects are quite acute, some of the effects are quite chronic. But you've asked an important question, actually, because you can probably appreciate that these are very acute experiments, whereas actually amphicycline-related toxicity is a chronic problem. Our argument is you really need to take this sort of aggressive approach to reveal some of those fundamental mechanisms.
We've got a question from somebody listening via Teams or online. Is this why it's important to have calcium in your diet?
So yes and no. So dietary calcium is important for all sorts of reasons, but the levels of calcium we're talking about here in terms of heart cell contraction are absolutely tiny. So if a lack of dietary requirement, calcium started to cause you problems, you'd be long dead before it affected your heart. It's a different sort of order of magnitude, but it's a great question. But obviously it's important in the long run. Fantastic question. It's on the list. That's Beth and Alicia are in the middle of doing that as we speak. So they're looking at mitochondrial function. We're pretty sure. I didn't have time to go into all the details. What we do know is that oxidative stress is coming as a result of two enzymes in the cell. One's NADPH oxidase and one's xanthine oxidoreductase. What we haven't ascertained yet is whether the mitochondria also contribute to that ROS production. And it's almost certainly the case that they are. It's a great question, yeah.
Well, if there are no more questions, my last very pleasant duty is to thank Dave. I think we can all see why you've been appointed as a professor. I mean, not only do you do world-leading research, you're clearly, as nobody can be any doubt, having listened to you, a very enthusiastic teacher. And I found the beginning part of your lecture talking about sort of social deprivation, access to university, really, really thought-provoking. So thanks very much.
Pleasure, thank you.
Thank you. Finally, we have a little memento here. I suppose I should ask the audience, do we think he deserves it? Okay. On behalf of the University of Salford, presented to Professor David Greensmith on the occasion of their, very politically correct, inaugural professorial lecture.
Thank you very much. Thank you. That's great. Thank you.
Now I think it's next door.
The bar is open. Thanks, just open.